Thank you very much for having me, Kathrin. It's really lovely to be here, and thanks to everybody for joining us on this Monday evening, for me at least, and I think even later for a lot of you. So like Kat said, I'm a biomedical science advisor at the moment with the Humane Society of the United States and Humane Society International, which is an animal protection organisation. And the AFSA collaboration and Biomed 21 that Kat mentioned are like the science arms of that animal protection. And AFSA is focused on animal-free chemical safety assessment. Biomed 21, which is where I work, is looking at animal-free biomedical research. So we're working with a range of stakeholders, with the researchers, academics, funders, regulators, just everybody that's involved in creating better science to try and accelerate that move away from animals towards more human relevant models of science. And like Kat just said to you, I wasn't always an animal protection person. I was a proper researcher, so I did actual experiments, and I'd like to take you back in time a little bit to talk about that. And that was my research modeling cystic fibrosis. So in order to do that, we're going to have a quick look at the human airways. What do they look like and what do they do? Then I'll go through some of the data that we had collected using human relevant models of cystic fibrosis. And then we'll look beyond that, because in 5 years, a lot has happened for human relevant modeling. So we can see what's developed since then and where might we apply it. And obviously, if we're talking about airways, we can't not talk about COVID-19. So we'll have a look at how some of these tools are being used for COVID-19 and how we might use them more widely than that. So if we start with a sort of a glimpse of the human airways, we can see that they are pretty complicated organs. They're not just about breathing, they're much more than that. There are these distinct regions within the airways that are made up of different cell types and have relatively different functions, really, and that's linked to the different cells that are found in those different areas. So the nasal cavity is really good at filtering out particles, but maybe not so good at gas exchange, obviously. The trachea and the bronchus are really good defensive regions where they're trying to maintain sterility. And as we get further down, we get to the alveoli, which is where we find the gas exchange. So if we think about the respiratory tract as a whole, we can see that it's not really just doing one function. There are many different areas that are doing these different functions. And when we're thinking about research, that's really what we need to think about. So modeling these airways, you can see, is equally complicated. And I can't really see or think of a reason why we'd want to model that entire tract from the nose all the way down to the alveoli. If we think about what's in there, there are enough cells to cover the surface of a squash court. And what we've done in the human airways is take all of those cells and kind of scrumple them all up together and put them in a 1.5 litre container, which is impossible for cell culture, isn't it? For an in vitro model, we're not going to get anywhere near that. So let's just be sensible and think about what we want to ask. Think about the questions that we are putting to our different models, and develop our models so that it's complex enough to answer the question, simple enough for us to understand, and really fit for purpose for what we need it to do. And if we're talking about fit for purpose and we're talking about humans, then I totally agree with Kat's boss, Thomas Hartung, that, well, Kat's former boss when she was at Johns Hopkins, that a human is not a 70-kilogram rodent. And if we look at the difference in airways between mice and people, we see some significant differences here that mean that actually looking at a rodent doesn't give us a particularly great insight into what might happen in a person. So I've just picked out some features on this table here. This is not an exhaustive list, but these are some of what I feel are the more important features when we're thinking about modeling. The first is that mice are obligate nose breathers. People can breathe through their noses or their mouths. Now, maybe that doesn't have a great deal of significance until we think about maybe cigarette smoking or e-cigarette smoking. And then we see that actually people are using an entirely different mechanism almost to get that smoke into their lungs compared to a mouse. So if we're looking at an animal model of smoke exposure, we need to do either whole body exposure, where the animal's stuffed in a box and covered in smoke, or we plug something onto their nose and force it into their nasal passages. And that doesn't exactly recreate what people are doing. And I guess the other difference, if we're thinking about breathing patterns, is that for a mouse model, we're looking at when the staff are available to expose that mouse to smoke, which tends to be 9 to 5, Monday to Friday. Doesn't exactly model when people smoke. So we've got an issue there as well with just that kind of technological problem. If we're thinking structurally, the branching patterns of airways are very different in mice compared to humans. And that has ramifications if we're thinking about drug delivery and airflow and where different sized particles might deposit. So it's difficult to look at what might happen in a mouse airways, which has this sort of monopodial pattern where we've got one branch coming off, compared to the human airways, where we've got dichotomous branching. So we have two branches popping off. And we've seen, or I've mentioned, that the different regions of the human's airways have different cellular compositions linked to their function. And that's kind of true for the mice, but also mice and humans are very different. So it's kind of not always possible to take a region of the mouse airways and histologically match it to a region of the human airways, because they don't just match up. There are different cell types and different cell densities in mouse airways compared to human. So they're just different. And there's been recently, Mark Davis from Stanford University has been really pushing for a move to use humans for immune responses. So looking at immunology research. And there's a huge body of data for mice, but again, it doesn't really map to humans. And as we start to build more human data, it just kind of makes sense to try and make that switch towards using that human data and stop relying on the mouse data, because it doesn't map on. And really that makes sense if we think about immune responses, because mice live in a very different environment to humans. So they need a very different immune response to humans. Mice don't really respond to lipopolysaccharide, which is one of the components of gram-negative bacteria cell walls, because they're kind of scurrying about in the dark and in the dirt, and they're full of LPS, basically, lipopolysaccharide. So they don't really bother about it. They don't respond to it. It's very different for a human. That's a huge alarm signal to us, and we would respond quite vigorously and try and clear it. So you see, we have these dichotomies here that mean that it's very difficult to say that one's the same as the other, because they're not. And the bottom line to all of this, and why we're kind of why we're doing this today, really, is that for SARS-CoV-2, mice are not naturally susceptible to this virus. They have to be manipulated in order to be infected with this virus. So they have to be artificially engineered to have the virus. And sadly, we have more than enough evidence to show us that that's not true for humans, and we are very much susceptible to this virus. So we have these huge differences between mice and human, and it's not just mice and human. It's pretty much every mammal that's used for respiratory research is not the same as a human. So sheep, guinea pigs, hamsters, ferrets are all used for research into respiratory diseases. And these are data in this figure here taken from the EU and the UK from 2019. And this is looking at animals that are recorded under the categories of either respiratory system research for basic research or human respiratory disorders. So I've combined those two columns, or those two categories. And we find that if we look at the EU, we've got about 140,000 animals are used just for these two categories of respiratory research every single year. For the UK, it's around about 20,000. And for each region, for the EU or the UK, we see that mice bear the brunt of this research. So despite those differences that we talked about, despite the recognition from eminent scientists that mice don't model humans, they're still used very heavily. Around 3/4 to 80% of animals used for respiratory disease research are mice. And this is despite the fact that mice do not spontaneously suffer from the main respiratory diseases of people: asthma, cystic fibrosis, chronic obstructive pulmonary disease, pulmonary fibrosis, or even COVID-19. There's no natural susceptibility of mice to these conditions. Now, it's not just me that says this. Of course, you're going to expect me to say this, given my role in animal protection. But this line here, these lines here, come from the papers from the researchers that are using animal models. So these are the people developing the animal models that freely acknowledge that there is no single model that recreates all the features of a human disease. So we cannot say that there is a mouse model of asthma, because it does not exist. And what happens instead is that animal models recreate one or maybe two aspects of the disease. So they might have some symptoms of the human disease. They might display one or two of the same mediators of the human disease, but nowhere is there a true avatar in the mammalian world that is non-human that displays the characteristics of these human diseases. And what I'd like to do now is go through my work looking at cystic fibrosis, just to give you an idea of where we were developing these human relevant models using human cells, and what we used them for, and how we can actually start taking this reductionist in vitro approach to be able to get some fairly sound answers, really, for what this disease is and how it progresses and how we might start to address it. And my apologies to any of you who are involved in cystic fibrosis research right now, because as I said, it's been 5 years since I was in the lab, so this might all look hugely dated to you, but I still think it's really exciting to see what we can actually do with these, with these models, excuse me. So if we think about cystic fibrosis, then this has been around with us forever. It's a monogenic disorder, and the European folklore said that the child with the salty brow shall die, which is quite, you know, it's a horrible sort of statement, but it indicates the prognosis for this condition before we really understood what it was. And the salty brow came from the inability to reabsorb chloride, because this disease is caused by mutations in the gene encoding the chloride channel, CFTR. So we get aberrant chloride conductance, which leads to excess chloride in sweat, also pancreatic insufficiency, which is addressed to the main with digestive enzymes. And what's most severe in cystic fibrosis is the lung disease. So lung disease kills around 90% of the people that are born with cystic fibrosis, and it's a horrible progressive lung disease that's linked to this thick mucus in the lungs, which just invites infection, and there's chronic infection that just is impossible to clear, really. And on top of that, there's a relatively poorly understood hyperinflammatory response as well. So people with cystic fibrosis tend to release more pro-inflammatory mediators that just exacerbates this condition, really, and drives the tissue damage. So this is, I couldn't talk about cystic fibrosis without showing you what CFTR looks like. So this is the channel that is mutated in cystic fibrosis. It's an ABC protein. So for those of you who are protein biochemists, it's an ATP binding cassette protein, and it binds ATP on its nucleotide binding domains, of which it has 2. And that can result in phosphorylation of this regulatory domain, which kind of blocks the pore, or when it's phosphorylated, moves out of the way, and we create a hole in this channel. And that hole, the pore, is where we can allow chloride to transfer. And the chloride movement drags water with it. So water can either move paracellularly or through aquaporine water channels. And what we get then is hydration at the apical surface. So this is an apical membrane channel. So that movement of chloride pulls water through, and we get this fluid layer at the apical surface. And that's really important, because this is my own cartoon. I'm sure you can tell that I drew this myself. And this illustrates what's going on in the airways of somebody with cystic fibrosis. So on this side, on the left, I'm hoping that you can all see my laser pointer. We have our epithelial cells with cilia on the top of them, and the cilia sit in this liquid layer. So that liquid is the consequence of CFTR activity and the water, and the cilia are happy in the liquid layer, and they can beat this overlying raft of mucus towards the back of the throat to dispel it from the body. And that's our mucociliary clearance, which is one of our great defense mechanisms. That means that whatever we've breathed in, we trap it in the mucus and we get rid of it. Now, if we look at what happens in cystic fibrosis, we see that this has all gone wrong, basically. And now we have a lack of chloride transport, which means our water movement is reduced, which means the periciliary liquid is reduced, which means the cilia are stuck and they can't beat anymore. They're just kind of got their heads basically caught up in this dehydrated mucus, which invites colonization by bacteria. The bacteria get in there, they can sit in there, they're quite happy in there. They can form their biofilm, and they can just become this horrible, long-lasting infection. And the fact that the cilia can't beat means that we can't move that mucus out of the lungs. And you might already be aware that people that have cystic fibrosis have to undergo quite strenuous physiotherapy on a very, very regular basis to try and shift this mucus physically, because this physiological mechanism is no longer working. And it leads to this cycle of damage then. So we see this sort of knock-on effect of our CFTR doesn't work. We get conditions that mean that we get colonization by these opportunistic pathogens, I have to say. That leads to inflammation, which never resolves because the pathogens are already there, always there. The epithelial cells are kind of calling for help, releasing these pro-inflammatory mediators, our friend interleukin 8. This is a potent neutrophil chemoattractant. So the neutrophils will pour into the airways. And in cystic fibrosis, the neutrophils are already primed. So they're already a bit excited and a bit spiky. And as a consequence, they're very free to release their proteolytic enzymes like elastase, which can have a knock-on effect and damage the airways tissues. And as this progresses, we get into this cycle, which is never broken because we've always got the stimulus. So we've always got the response. Then we get ultimately damage to the airways tissues, which leads to bronchiectasis, narrowing of the airways, and ultimately, unfortunately, it's the lung disease that will be fatal for people with cystic fibrosis. Now, that all feels very gloomy. So I'd just like to say that things are getting better. And in 1989, when the gene was first discovered, life expectancy was probably around 29 years. As time has moved on, you can see this is median predicted survival from the US Cystic Fibrosis Foundation. We can see that we've had a nice steady increase in survival. Now, obviously, it's still not good enough, but as of now, median survival is around 47 years. And some people, it's shocking to say it, but some people are even living to beyond 50. And it's shocking because it seems horrible to say that out loud, doesn't it? But that's a huge milestone that we've got. People with cystic fibrosis are surviving that long. And that's partly because we screen much more effectively now, and partly that we've just recently got some new treatments that seem to be really improving the prognosis. So if we skip back to here, it's not about identifying the gene and developing animal models of this disease that has really helped us, but actually it's much more screening, improving our ability to spot those people that have got the disease early and be able to go in and treat if we need to. And the new treatments are hugely promising. So this is not my data, or these are not my data. These are, I think it's BBOC. I can give you resources afterwards if anybody's interested. But these are using small molecule modifiers of CFTR. And ivacaftor was the first one that we heard about, or its trade name is Kalydeco, and it is a potentiator. So what it does is maintains that open state of CFTR once it's open, so it can permit more chloride movement, which leads to more water movement, which allows an increase in the periciliary fluid layer. And what we see here, so I haven't really mentioned this, but there are thousands of mutations in CFTR that can lead to disease, and different mutations lead to different severity. This one, F508 deletion, is the most severe mutation. So homozygous for this mutation is the worst prognosis, really. And we see that actually this potentiator doesn't do a great deal of improvement for CFTR activity for this mutation. And that's because the CFTR protein with a deletion of phenylalanine in position 508 gets just lodged in the endoplasmic reticulum. It can't be processed properly, and it doesn't ever make it to the membrane. If we can push it to the membrane, we can get it to be active, and that's kind of what this is suggesting here. But really, we need a different strategy for this. And where we see Kalydeco working more effectively is people that have this G551D mutation, because this protein is in the membrane, but it just has a defect that doesn't allow it to gate properly, so it's not letting the chloride out. And then, even though this is homozygous for G551D and the F508 deletion, we can treat it with the Kalydeco and massively improve CFTR activity. So these drugs have been a huge game changer for people with cystic fibrosis. And we see that activity changes, but then look, we can see the functional impact of increasing activity. And this is measuring the depth of the airway surface liquid, or that periciliary fluid. So we see that this increases in the presence of ivacaftor. And then if we use this compound that increases fluid secretion in the presence of functioning CFTR, increasing ivacaftor increases that volume of airway surface liquid. So we can see now that we have a real strategy to be able to reverse that ciliostasis and try and maybe kickstart the mucociliary escalator. And these drugs have been just incredible in transforming the lives of people with cystic fibrosis and actually starting to improve their lung function, which is what we want. So we want to not just stop the damage, but actually try and reverse it and get lung function back to as close to normal as we can, and allow people to live a healthier life for longer. And why I wanted to use this example is that these small molecules, the efficacy testing for them was carried out on a broadly in vitro platform. So you might already know that for drug development, there are elements where animals are used for safety testing. For some reason, the regulators still require whole animal testing. But here, this is for efficacy. You could not use a CF mouse to show that this potentiator would increase chloride movement through CFTR, because the mouse lungs don't really care about CFTR. They're absolutely fine. Mice don't get CF mice don't get the lung disease that typifies the disease in humans. The first cystic fibrosis was created a few years after the gene was cloned. So this was, you can imagine, this was heralded as the greatest advance. We had the gene that caused this horrific condition that was causing people to die before they were 30. So this seemed like, okay, we've got it now. We created the mouse in 1992, fantastic, now we've really got it. And 30 years later, we still don't have any curative gene therapy. Now, there are many issues to this. It's partly that we're talking about lung cells, which tend to have quite a high turnover. We've got that awful mucus in there, but actually it's just proved really difficult to get the CFTR gene, which is massive, get it packaged up and get it into those epithelial cells. And all these CF mice, the hundreds of models of them there, have, well, I'd say have not got us the gene therapy. There have been efforts to produce CF pigs as a larger animal model that might recreate the progression of the disease, but to be honest, I haven't seen anything fantastic coming out of those. CF ferrets, CF sheep. So maybe some misguided attempts to try and understand the disease by looking at larger animals or different animals, and not a kind of a desire to just take a step back and think, what are we really thinking about? And with cystic fibrosis, there are, I think, around 2,000 different mutations, of which, of course, you can have heterozygotes and homozygotes, and they will all of them have different disease severity, and that the CFTR itself is a modifier gene, so it has knock-on effects on other genes. So it's not as straightforward as we maybe first thought when we identified that gene. And the issue with some of these mutations is that they'll be quite rare. There might only be a couple of people that have that, and maybe we haven't even discovered all the mutations yet. So hopefully you can see that perhaps it's not feasible to think about creating a pig and a mouse and a ferret and a sheep that has this mutation. And perhaps it is more feasible to think about, well, what can we do with human cells? How can we use those to start to look at the rare mutations and how that might impact the disease, and how we might start to address that? And my final point is, this comes from a clinician that I used to work with, who said that, and he was a clinician who was a pulmonary scientist, so he was interested in his patient's lung function. And his statement was that the CF mice, they might recapitulate the intestinal blockage that we see in babies with cystic fibrosis, but they're not any good for lung function. So let's have a look at something else then. Let's have a look at using humans instead. And these are a couple of histological images of human airways epithelium. So on the left is our healthy non-cystic fibrosis epithelium. So these are our epithelial cells at the top, sitting nice and tightly together. And you can see here some cilia on their apical surface. So this is the air side, and this is inside the body. So that's all quite beautiful. And that's possibly a bit of a drift of mucus in there. But when we compare it to our cystic fibrosis airways epithelium, all of this purple gubbins here is mucus. So that's what we're dealing with when we're looking at cystic fibrosis. You can see the epithelial cells are pretty much swamped by that mucus. It's just all pervasive and invading and thick and sticky and horrible. And this is what we want to model. That's what we want to see. So how do we go about doing it? Well, you'll laugh at this if you have anything to do with cell culture, because this was revolutionary for me, because inserts came into play and transformed our cell culture from 2-dimensional to 3-dimensional. So prior to this, we'd been culturing our cells on flat plastic flasks like this. So the cells were just completely submerged in liquid. And even airway cells would try and be airways. They try and kind of sit together and form sheets and barriers and do the things that airways should, but we're asking a lot of them to do it on a submerged, effectively drowned culture. And bringing these inserts in that allowed us to put cells on here and have them fed basolaterally was game-changing, really, I think. And this is what we set up in my lab. So we had a core culture model, so it's still quite reductionist and still incredibly simple, but we thought it's important to put fibroblasts in there because we know that they're involved with repair, and we know that they've got a role to play in releasing growth factors and also supporting cells, and kind of, we just thought that we wanted to do something beyond a monoculture now that we had these the ability to try and mix cells together. So we would take pulmonary fibroblasts, put them on top of this insert and give them a couple of days to kind of settle down and get themselves sorted out. And then we would take our airways epithelial cells. For cystic fibrosis, we used a cell line because at the time I didn't have access to patients. And we would put the epithelial cells on top of the fibroblasts and leave them for a few days with medium on their heads at this point, so they were submerged. And then after 4 days of submerged culture, we remove that apical medium and we have cells at what we call air-liquid interface. And the magic of the epithelial cells means that under those conditions, that's the only driver they need. We didn't have to add any growth factors or any specific stem cell factors or anything complicated like that. We can leave them at air-liquid interface and they will differentiate beautifully. And we could see that we had a basal population, and then we had our pseudostratified layer with cilia on the top and with goblet cells, so they're making mucin as well. And I have an electron micrograph of these. Don't laugh at these. I know they're not particularly beautiful, but we thought they were fabulous when we made them. And our C38 cells is the corrected cystic fibrosis cell line, and IB31 is the cell line that maintains the mutation. So IB31 has an F508 deletion and a missense mutation, so it's a heterozygote line. And hopefully you can see from here that this is the membrane, and then these are our cells kind of piled up on top. So we have a semblance of this pseudostratified layer. You know, we haven't just got one layer of cells, we've got this stratified layer, and we can stain them, and we can show that we've got the basal population by using cytokeratins, and we've got this differentiated population. And if we look at the brown staining down here, this is for our fibroblasts. So we see that our fibroblasts do stay nearer the membrane, and our epithelial cells kind of sit on top of them. And that's what we wanted to see. So we say, okay, tick number 1, we have a pseudostratified epithelium. Tick number 2 was whether or not this was functional at all. And given that our airways have to be a barrier, then we wanted to look to see if our airways models were a barrier. And we did that by looking for tight junctions. So the tight junctions is what polarizes an epithelium. So it maintains the distinct compartments of the apical at the top and the basolateral at the bottom, and they kind of form a ring around the cells and allow distinction so that proteins cannot move between apical and basolateral. And zonular occludens 1 is one of the main tight junction proteins. So we used immunohistochemistry to stain for that. And actually this is confocal microscopy. So what we could do was look upwards through the cells. So if we start at the bottom of the cell, when we're closest to the membrane, we see that we don't really have a great deal of ZO1 staining. And hopefully you'll see the difference, because this is looking at the apical surface of the cell. And now you can see that our cells are surrounded by green, which is an indication that ZO1 protein is there. So at the upper surface of our cells, we have this tight junction protein that's maintaining our apical and basolateral differentiation. And that's functional because we can measure the electrical resistance. So TEER tells us that we have a barrier. High TEER is a tighter barrier. And this is a really straightforward way to measure that we've got a barrier. It's quite easy. And it's non-destructive in that you can do multiple measurements of TEER without damaging your cells. So you could measure this every single day, which is what we did for a long time. And we see that if we leave our cells at submerged culture, so when they've got liquid on their top and on their bottom, they don't create this tight barrier. There's no drive for them to do that polarization and electrical resistance, really. But if we take the medium off the top, then what we find is that we get an increase in this barrier over time. So we have tick number 2 for our physiology, that we've got a barrier function. But actually the other thing that TER gives us is an ability to have a look at kind of the health of our barrier. So if our TER was dropping, then we knew that something was wrong. And as I said, it's really easy to measure this. We just use a chopstick electrode in the insert and outside of the well, and we get a reading. So we can do it as many times as we like. And that's kind of difficult to do in living mice, at least, or living people even. But certainly for our model, it gives us an advantage that we have this real quick snapshot measurement of how are our cells, do we still have a barrier, what's going wrong? So we have that ability. We've talked about the importance of kind of mucus, not just for defenses, but also in disease as well. And we looked at our model to see whether or not we were releasing mucus. Now, I can appreciate that there are a lot of boxes on here, but if you concentrate on the ones with the black bars around them, those are our apical supernatants. So that's when we take what's at the top of the cells, and that's where we'd expect our mucin to be secreted. And that's what we find. So we have our normal airways, so without the cystic fibrosis mutation, and then we have our cystic fibrosis airways here. This is A and B are with fibroblasts, so that's epithelial cells and fibroblasts. When we look at epithelial cells by themselves, which are these boxes, C, D, E, and F, the black boxes, we see that the epithelial cells are making the mucus, which is what we'd expect, really. If we look at fibroblasts by themselves, which is in H, they're not creating any mucus. So I know that's probably not a surprise to you, but we just had to make sure that, you know, there's nothing freaky going on here and our fibroblasts have suddenly de-differentiated into epithelial cells, but that's not the case. And what we see is that our epithelial cells, whether they're normal or cystic fibrosis, are both creating mucin. this was stained using MUC5AC, which is the major mucin in airway secretions. And this secretion is directed preferentially towards the apical compartment, and we don't find mucin in the basal compartment. So that's another tick for our functionality, that we're getting mucus released apically. And in terms of our functionality then, let's have a look at the other thing that we're interested about in cystic fibrosis, and that's interleukin 8. So this is our neutrophil chemoattractant. And what we see here is that our cells are releasing this chemokine, but that it seems to be directed more towards the basal compartment. And that kind of makes sense if you think, well, our neutrophils are actually in the basal compartment. If you imagine that this was an intact body, the basal is, you know, the blood side. So maybe we'd want IL-8 going in that direction to kind of signal to our neutrophils that it's time to come out and do something. And I think what this really illustrates as well is just how straightforward and easy it is to get access to both those compartments. So we can take what's coming out at the apical surface, and we can take what's coming out at the basal surface, and we can do that as often as we like over the course of an experiment. And we did track the release of IL-8 every couple of hours or so, just to see what was happening in the presence of pathogens or without. And that's something else that I don't think it's really easy to do, maybe in an animal model or in a more complex setup. Certainly in a living animal model, I'm not sure that you'd be able to have access to these compartments in such a simple manner to be able to do this sort of analysis. So it's one of the powers of this model, is that we can look at what's happening and where it's happening and when it's happening. And if we get a bit more specific now and think about, okay, what about cystic fibrosis? Those features that I've shown you all seem to be quite generic for airways. Well, we can show that the model models the hyperinflammatory response. So we have our non-cystic fibrosis cells that, under control conditions, they're releasing a bit of IL-8. If we hit them with a pathogen, then they release a bit more IL-8. Our cystic fibrosis cells, interestingly, we've got the same sort of baseline levels of IL-8, which is kind of what we'd expect. But look at what happens when we hit the cystic fibrosis cells with a pathogen. We get over twice as much IL-8 released. So this is an indicator to us then that we've got a hyperinflammatory response modeled in this system. And it's really important that we can see this IL-8 response, because that's what a lot of people are trying to target as a key mediator of the lung damage that we see in cystic fibrosis. And as a bit of an aside, mice don't have any IL-8. So we couldn't do this sort of analysis. We could look at equivalent or similar chemokines in mice, but it's not IL-8. It's not exactly the same. It doesn't have the same features. So that's something human relevant about our human model. And this is something that is just as human relevant, I think, and that's thinking about host pathogen interactions and thinking about how specific they are between hosts and pathogens. And for cystic fibrosis, the issue is that these opportunistic infections come in and create such damage. So an opportunistic infection, I'm sure you all know, is something that basically it wouldn't bother a non-cystic fibrosis person. So we could swim in a bath of Pseudomonas aeruginosa. It's found in the soil, it's an organism that's everywhere, and it's not really going to bother us. But if you get cystic fibrosis with the issues that people have in their lungs and their inability to clear the mucus and other defensive problems that appear to exist, then Pseudomonas aeruginosa becomes a huge problem. And we looked at Staphylococcus aureus, which is also present as one of these kind of horrible cocktail of microorganisms that bedevil, I guess, people with cystic fibrosis, Pseudomonas aeruginosa and Burkholderia cepacia. So these are three of the big pathogens that we'd be worried about in the clinic for our patients to turn up with. And we looked at our non-CF airways model in the white bars here. And what we did was put these different bacteria on top of our model, left them for a couple of hours, washed them off, treated with gentamicin to try and get rid of the remaining bacteria, and looked at how our epithelial cells have fared. So the cell viability here is looking at that epithelial model to see how does it manage with these different bacteria. And the non-CF models don't really care, fine, not bothered, as we'd expect. Opportunistic pathogens, they've got enough defenses or whatever it is they're doing to be able to protect themselves against it, but these bacteria in that short-term infection model are not impacting the cell viability. And yet the story is very different when we look at the cystic fibrosis model. And then we see, under the same conditions of infection, a much more deadly impact on our epithelial cell layer. So we have a reduction in viability to sort of 70% and down as much as 60% of our baseline levels. So these CF pathogens are destroying our CF airways. And we think, okay, that's a good sign maybe, because maybe we can start to look at why is that happening in CF and not non-CF, and maybe we can start to work out why are these opportunistic infections no longer opportunistic in this particular model. And this is a very useful and convenient way to be able to look at that. So we can look at pathogens, but we can also look at treatments too. So this is quite busy, and I think I'm not really bothered if you don't really get all of these bars on here, but let's just concentrate on the overall aims of what we're doing here is to take our airways model, and we're going to put bacteria on there again, and that's what we do in our control. So we put the bacteria on there, and then we leave it for a couple of hours, and then we're going to treat it with a potential treatment or not, and then we're going to take off the bacteria and count them again, and that's what we've done here. So we count our bacteria to see what has happened to them, and what we're looking for is whether our potential treatment can reduce bacterial numbers. So our control, that's our positive control that says, okay, we get over 10 to the 9 colony forming units per well when we don't do anything. And if we start to look at our different treatments, you can see that we can start to see which of these are capable of reducing the number of bacteria on our epithelial cells. And what we were interested in here was whether or not we could use something like lactoferrin, which is a component of the innate immune system. We're interested in lactoferrin because bacteria cannot derive resistance to lactoferrin. We know that we've got a huge issue with antimicrobial resistance and antibiotics we have to be so careful with. And, you know, I think we've seen, I don't know, some terrible stories about things not working anymore. So we really need to, okay, let's stop and think about this. And our hypothesis was that, well, maybe we could start to mix antibiotics with more natural defenses to see whether we can maybe reduce our reliance on antibiotics. And that's what we were doing. We could show in this model is that we could treat our infected airways with these different potential treatments and then look to see which ones were more effective at reducing bacterial numbers. And what we see is this tobramycin is an antibiotic that's used quite frequently for cystic fibrosis. So it's exciting to find that we can spray dry tobramycin to deliver it straight into the airways, and it's still very effective. And we can mix it with lactoferrin, and maybe there's a hint more effectiveness there, maybe there's not, but there's certainly, we didn't carry this work on because I'd left by then, but there's certainly potential to look at titrating the concentrations of antibiotics with and without these native proteins to see whether or not we can get the ideal combination. And again, our model gives us a really straightforward, simplistic platform to enable us to do that. And it wasn't just kind of potential treatments we looked at, but we couldn't resist having a look at the Vertex drugs, because as I said, these Vertex small molecule CFTR modifiers were just appearing as I was sort of leaving academia. But we did get our hands on some VX809 or lumacaftor, which is a CFTR, it increases the trafficking of CFTR. So remember I said that the F508 deletion mutant, the protein gets all blocked up in the ER. Well, with VX809, the thoughts is that it will be pushed to the apical membrane. So we use that on our IB3 model. So if we look at the apical surface of our CF cell model at baseline without any treatment, we can see that we've got kind of a purple haze, which indicates that we've got very low CFTR expression because our CFTR is green under these conditions. If we treat our cells with VX809, the lumacaftor CFTR promoter to push the CFTR to the apical membrane, then that is what we see. So our model is responding to that drug exactly as we'd expect us and gives us a really good, clear readout that expression of the protein has been moved to the apical membrane. So we can see it there. So that, you'll be pleased to hear, is the end of my whistle-stop tour of my life in cystic fibrosis. And what I'd kind of like to think about now is what else can we do with these NAMs? So we call them NAMs, the new approach methodologies or non-animal methods. And I think even in the 5 years since I've left academia, the evolution of these models has been impressive. It's just been hugely exciting to see the things that we can do now that we couldn't do when I was in the lab. I left before the lung chips even appeared that I'm going to talk about. So I feel like it's just a most fast-paced field, really, to try and keep in touch with it. And underpinning this is, of course, if these NAMs are getting so good and evolving so quickly and being so useful and we're able to exploit them effectively, then we don't need to use mice or monkeys or hamsters or dogs or anything else. And I'm sure you've probably all heard of these already, and you'll be glad to know that we're not going to go through all of them now. But I'd like to consider some of the advantages of these models of the human airways, and just to kind of think about, well, why these are maybe quite often a better route to get the answers to the questions we're asking than using animal models, honestly. And I think the first huge advantage is that we must remember that the species of interest to us is human. So it seems to me hugely important to use human cells where we can, particularly if we're looking at infectious disease and host pathogen interaction, and we see that we get that, you know, really exquisite relationship between a pathogen and a host that is very difficult to recreate. We see it with the Pseudomonas and cystic fibrosis mice. The mice do not harbour those bacteria in their lungs and do not get infected by it. The same for SARS-CoV-2. So if we're talking infectious disease, let's talk about the right host and pathogen. I'm hoping that I've shown you from just the kind of fairly simplistic work that I did, that these models of the airways allow us access to different compartments. So not just within that cell culture, but also within the airways. We can set up nasal models or small airways models or bronchial models or alveolar models. And it's kind of difficult to do that real time in a living animal, but it's really straightforward to do it in vitro in these sort of culture models. And given that these are so small, these inserts have a surface area of about 1 centimetre squared. We can use tiny quantities of drugs. So if you have an experimental drug, you don't need to use all of it just to see if it's going to work, but you can use a fraction of it to look at efficacy before you move forward with, you know, anything else. So they're economical. We can sample as often as we like, really. A lot of the methods that we use are non-destructive, and a lot of the evolution of these methods now means that these can be sampled in real time. So we can put sensors in there to measure glucose or oxygen or transepithelial electrical resistance. So we can be continually monitoring what's going on, which is amazing. We can view them real time, so we can do fantastic microscopy, we can reuse them. And not all of these features are possible in an animal model. So I think let's consider what the question we're asking is and work out how we can start to use these human models. And there's more, we have more advantages of these human relevant culture models. And that's that we can be as simplistic or as complex as we want. And that's something that Don Ingber, who's head of the Wyss Centre for Biological Engineering, as I'll talk about in a couple of slides' time, is very keen on. It's about developing the right model for your purposes. So yes, of course, my tiny little snippet of airways doesn't resemble a whole human person with cystic fibrosis, but it told me an awful lot about what might be going on in that region of the airways in somebody with cystic fibrosis. And that's kind of important in trying to tease out what's going on in this disease. And I could see what the fibroblasts were doing. I could see what the epithelial cells were doing. There have been models that include platelets or neutrophils or macrophages. So we can look at how do our immune cells respond. So we can build these models as complex as we need them. And I think that's the crucial point. We can use cells from individual people. And that's maybe not so much for cystic fibrosis, to be honest, in terms of it hasn't been used a great deal for cystic fibrosis. But I think in terms of cancer, we're doing this a lot. We're doing it incredibly effectively to look at that individual variability between tumours. That's become a really good tool that I think we could exploit more effectively to look at really effectively treating cancer. Our cultures are amenable to genetic manipulation as well. So we hear a lot about CRISPR-Cas9 in animals. We can use it here as well. Don't worry, you don't have to discard your CRISPR-Cas9 if you want to use in vitro models. We can still do it here. We can use it to knock in genes, maybe, if that's what we're interested in. We use it to correct. If we have a cystic fibrosis model, we could correct it with CRISPR-Cas9 and then we've got our control. Or we could use it to knock out genes and see what happens. So we can still do a lot at this level, and I guess we might have fewer off-target effects if we're doing CRISPR in just one particular cell type. And then I think it's important to mention ethical. Obviously, I'm coming from an animal protection background. I think it's really difficult to make the argument about an ethical use of animals in research and testing, because I don't believe it is necessarily ethical. And yet I would say that the use of these in vitro methods is ethically sound, can be ethically sound, and you do not need now to jump through a million flaming hoops to be able to use human tissue, but there are many companies that are set up that have commercially available either cells or actual inserts, so cells at air-liquid interface. So it's really easy for you to be able to access these cells and to be able to use them. And you don't have to have a connection with a hospital or a clinician or something like that, but it's much more straightforward to get your hands on these cells. And if you're doing in vitro methods anyway, then it's not a great step to move to these sorts of models. It's not like you have to get a license to be able to use them, or you need a project license or, you know, institutional approval. You can just switch to it. And if you've already using in vitro methods, you've got the kind of cell culture and everything set up there. So I don't think it means a huge outlay on masses of expensive equipment for you to be able to do this sort of technique. So I'm just going to have, I've only got a few slides left, so if you bear with me, I'd just like to think about what we can do with NAMs, and then let's have a quick look at the lung chip, which is my favorite NAM, and some of the things that have been done with a lung chip. So I think really the question here is, what can we not do with NAMs? I honestly feel like these have application for a whole gamut of diseases that we're interested in, and we can exploit them in various different ways to look at toxicity, to look at different exposure scenarios, to look at different methods of drug delivery. We can start to model rare mutations. And I think that's an important point, because often for a human disease, there may not be enough patients to kind of collect enough people for a clinical trial. But if we're smart, maybe we can start to develop enough in vitro models of this rare mutation to get an idea of, okay, how would we move forward with a clinical trial? And I think it just seems more accessible to me to do that in an in vitro setup than it does to try and set up the animal models of that mutation. So I think there's a lot of power with what we can do with NAMs. And this fanfare is the lung on a chip. So this is, you see, this is the chip, and within these channels, there are lung cells that we'll see a picture of in a minute. And you can see it's all kind of connected up because these are dynamic systems. This is the Wyss Institute's lung chip, which won an award in 2015, and I think is on permanent display now at the New York City Museum of Modern Art, which is pretty impressive, really, because it's really science meeting art here. So I think this is maybe a bridge that we can build between science and art. And this is what Wikipedia defines organs on chips as, for those of you that are not kind of aware of organs on chips. And you may know them as microphysiological systems. I think maybe that gives you a better clue as to what they are. And it is just a miniature recapitulation of an organ, or a kind of a section of an organ. So we're not talking about an entire lung here. We're not talking about all the alveoli and everything, you know, like a little tiny lung. We're talking about the resemblance. We're talking about a section of that organ with organ functionality. That's the important point here. And this is what Wikipedia says. So this is the promise of organ on chips, is obviously they caveat it very carefully, but perhaps they will abolish the need for animals in drug development and toxin testing. And it feels like they're starting to do that, but we have to push a bit harder to actually get them there. This is, I think, revolutionary, because it's not just a lung model, but it's a breathing lung model. And I think this shows us the potential of these chips. And that's that it's not just about a static, here's a lung, here's some epithelial cells, let's plonk something on the top of it. But what they've done here is put the lung cells on the top and endothelial cells on the bottom. So we have an air channel and we have a blood channel. This is a flexible membrane in the middle. And what you can do is apply force to that flexible membrane, and you can kind of push it to stretch it and contract it, and stretch it and contract it in a regular rhythm, exactly like breathing. And they've found that actually that stretch is incredibly important in maintaining the physiology of these airways. So this particular model was used to look at pulmonary edema, so using alveolar cells here, so the cells at the very bottom of your lung. And there had been some data from the clinic that showed that interleukin-2, which is used for cancer patients sometimes, caused edema. So it caused fluid to collect in the lungs. And that's quite distressing, obviously. And what's really interesting is that we could use the lung chip to work out not just the mechanism of this, but actually a way to address it. And that was only possible, or it occurred only in the presence of this dynamic breathing lung chip. So in a static system, the IL-2 didn't do anything. But in a breathing system, what they found was the IL-2 gradually did fluid secretion increased into this channel until this was completely blocked. And they think that actually the breathing, stretching, was sort of stretching the cells and maybe increasing the possibility of gaps and that was caused by the IL-2 and allowing that fluid to flow into the upper area. So the lung chip recapitulated the clinical picture. And then what's more exciting is that this is not my work, obviously. This is Dan Hu from 2012. They looked at what might address this. So found that angiopoietin 1 actually stabilized the endothelial junctions and could prevent that leakage in this breathing lung chip. So we have a mechanism for why this is happening, and we have a way to address it as well, using this fantastic in vitro model. It's not just chips. Chips are my favorites, but there are more models than that. And I would urge you, if you're interested, to have a look at this publication that was a project that I did in collaboration with colleagues at Ecomol and Professor Ian Adcock at Imperial College London, and with the Joint Research Centre of the European Commission. And we looked at thousands of papers from 2015 to 2019 that were using non-animal models to look at different aspects of respiratory tract diseases. And we found over 200 models being used. If you want to know more about them, please, I can let you have this link, but this shows you the gamut of these models. So precision cut lung slices, where are they being used, how are they being used, organoids, spheroids, stem cells, ex vivo. Honestly, it's really exciting. It's a really burgeoning field that is something that I'd love to still be in, I think. But let's finish just thinking about COVID-19. Okay, it's something that we can't not talk about, can we really? And the lung chip has proved pretty instrumental in some ways in looking at COVID-19, I think. And it's been used, in many different research groups, many different research groups throughout the world to address different aspects of COVID-19. So looking at the mechanism of viral entry, how does that, how does it work? What's important? So people have looked at models of different areas of the respiratory tract. Where is ACE2 expressed? And, how does that impact on COVID-19 research? So we start to work out that maybe it's this area that's important for viral entry, actually, because that's where the protein's expressed. So that's where the COVID-19 is getting in. How do the drugs have any sort of mechanism of action? What's going on there? How do we deliver the drugs? If we think about that 2-channel setup, is it more effective to inhale a drug and have it come down that air channel? What happens? Compared to having it systemically, so send it down the blood channel, what happens? There have been models that have put neutrophils in that bottom channel to see, well, what are they going to get up to then? And as I've already kind of mentioned, we can have a look at individual responses. And that's, at the moment, that seems to be done a bit more for cancer than it is for something like COVID-19, because I think we're all kind of scrabbling to just keep up with COVID-19. But that's an option for the future, is to look at these individual, almost personalized lung models. And I think maybe it's important to think for COVID-19 that, yes, this disease, I guess, starts as a respiratory disease, but then we know that there's involvement of multiple organs beyond that. So cardiac, brain, vascular. So linking a lung chip with a cardiac chip, the brain chip, the vascular chip, we can start to see the kind of, what's that organ level response between these different organs? How do they communicate? What's going on there? There's been some beautiful work with blood-brain barrier models to see, can SARS-CoV-2 cross the blood-brain barrier? And when it does, what does it do in there? That's all with in vitro modeling that is really quite fantastic. And I think probably in terms of the kind of kinetics of the response, it's easier to do that in an in vitro model than it is in an in vivo model. And looking, I guess, beyond COVID-19, we can think about, okay, what else might we do with these lung chips? What else might we do with these other NAMs? And I don't think any of these will be a surprise to you, because I think we've already really covered a lot of them. I've certainly alluded to a lot of them. Personalized medicine, we hear about that a lot, don't we? So taking stem cells from somebody and developing not just a skin model, but a brain model. It's kind of difficult to access brain cells from a living human, but we can do it with stem cells. So we can create a model of somebody's brain and their lungs and their kidneys and whatever we want. We can use that to look at rare mutations. We can use it to look at something broader like Parkinson's. We can use it to great effect with cancers. And there have been some clinical trials that have taken samples of the patient's tumor, grown it up in the lab for a very brief time, and treated it with different concentrations of different combinations of drugs, and then use that information to go back into the clinic to treat the patient more effectively. So I think that's pretty fantastic and is a real accelerated way to get from the bedside to the bench, back to the bedside again. So that works really well, I think. It's very exciting. I've already kind of mentioned that plugging things together and looking at crosstalk, and maybe this is really exciting for toxicity studies. So we can look at, if you put something on a skin model, well, what does it do when it gets to the liver or the kidney or the lung? So I think, you know, there are ways that we can develop these models much more to look at toxicity testing without using any animals whatsoever. It's more straightforward to use in vitro models, I think, for combination therapies, just because they're, you know, they're smaller, they're more economical. It's kind of easier to do that. And things like comorbidities. So there was a beautiful paper that looked at the smoking airway chip, and that was Camber's Benham in 2016 produced this, and took cells from people with chronic obstructive pulmonary disease and took them into the lab. And what they did was, for the same patient, they had the cells with COPD and then cells with COPD that were re-exposed to cigarette smoke on a regular basis. So they could look at what happens when you've got that continued smoking stimulus to these cells. And I just think that's quite wonderful. You can't do that with a human. You can't do that with anything, really, can you? You can't have, unless you've all got an identical twin, you can't have one of you stop smoking and the other one carry on smoking. But under these conditions, you can. You've got that kind of matched control. So you can look to see what's the impact of smoking here then. And what they find is that they got COPD-specific molecular signatures in their COPD patients, and that when they kind of kept smoking those COPD cells, they increased oxidative stress, as you might expect to find, and they drove ciliary dysfunction, again, as you'd expect to find in a smoking COPD person. So I think that has huge possibilities. We can do multiple infections. So I think we're starting to realize now that infections don't tend to be just one species of bacteria. Certainly for cystic fibrosis, that is not the case. And there's a whole hideous soup of bacteria in there. And I feel like these models offer us a really straightforward way to mix those bacteria together and look at what's going on, really. Disease progression. If we think about a lot of the diseases in terms of COPD, certainly, and maybe Parkinson's disease and things like that, they occur in later stages of life, quite frequently in humans. And taking cells from patients at an appropriate age allows us to see, I think, that disease progression more effectively than trying to stretch our animal model to as old as it possibly can be when it's just about to tip over into death and we haven't really got enough time to do our experiment. So I think maybe these in vitro models give us more scope to look at what's the difference between a young person with, I don't know, Huntington's compared to an older person with Huntington's, and see if we can start to look at what's the impact that age have on those diseases. And then the last example that I want to measure is just drug repurposing, which I think has been a huge success for COVID-19. And this is where a previously approved drug, so a regulator approved drug, is used for a different purpose than what it was already approved. And that uses a lot of computational modeling. So obviously the panic and the stress and the hideous nature of the pandemic really set people into motion to try and look at what do we already have out there, what looks like it might work against this SARS-CoV-2 once we worked out a bit more about that virus, and what do we have that we can kind of try and start to map onto things. So using computational methods to try and look at drug structures and targets and, you know, put those all together, look at in vitro models to see if we can do some preliminary testing to see what might happen, look at network analysis, for example, adverse outcome pathways. And I've got a quick plug here for the CHOW project, which is run by the European Commission's Joint Research Centre. And they're looking for people who are interested in doing this sort of network analysis where we're looking at existing data, the huge body of data that's out there for COVID-19. Let's try and organize it into an adverse outcome pathway to work out what networks, what relationships are going on between the virus and the outcome, the adverse outcome here, which is the lung disease or the cardiac disease or, you know, the endothelial disease, whatever it is we want to look at. Let's just try and start to map all that information together and structure it so that we can understand it. And please visit chowcovid.net if you're interested in learning more about that and would like to take part in it. And I think in terms of drug repurposing, then I feel like it's really promising that the one last example that I will keep you with is again from the lung chip, and that was work from Ingber again, so from the Wyss Institute, and they set up their lovely airway epithelial lung chip, their breathing chip, and they looked at 9 previously approved FDA drugs to see which of them might be able to stop the infectivity of SARS-CoV-2. And what they found was that, number one, they got different results in their dynamic system compared to a static system. So that kind of shows us that, you know, if we're moving towards these more dynamic systems, we're recreating that physiology more closely. And that appears to be a good thing to do, a more accurate thing to do. And number two, it flagged up this amodiaquine, which is an anti-malarial, as being able to inhibit infectivity by the virus. And I think that drug is now in clinical trial in South Africa. So this is an antimalarial drug. So it's already been approved. We already know it's safe. We already are aware of its dosing regime. So having this ability to look at, does it stop infectivity in a chip system, gives us a really fast accelerated push to go, yes, it does. Let's try it in a clinical trial. Let's try and give it to people and see if we can do something. So I think the issue is really, how do we use these human chips to increase trust and confidence? So trust for the regulators to be able to accept data from these, and instead of thinking that animals are the gold standard, let's try and turn it around and think that the NAMs are the gold standard. Let's not try and validate a NAM against an animal, because that's apples and pears and we're never going to get anywhere. And I just want to finish with a quote from, this is from Ingber's group, from their paper about amodiaquine. So they said, animal models are currently used as the benchmark for validation of therapeutics. They're not natural hosts for SARS-CoV-2. And when infection is possible, they commonly exhibit different systemic clinical manifestations from humans. So I think this to me just underpins the fact that we need to move away from trying to force these animal models to look like a human disease. And we need to really embrace the fact that the NAMs already look like a human disease. So let's try and exploit them a bit more cleverly and, you know, fit them together and make them work. And let's learn more about human diseases. And I would like to say a huge thank you to my former colleagues from Aston University, where I did the research that I talked about. Nanofarm, the Human Research Trust, and the Wellcome Trust were my funders at that time. My current colleagues at HSI and HSUS, Dr. Herrmann, for organising this and allowing me to talk to you all about cystic fibrosis for so long, and all of you for your attention. Thank you so much. My details are here. If you want to contact me, we'll try and answer questions now, but if you've got anything else you want to ask me about, please get in touch with me, connect with me, follow me on Biomed21, and thank you very much indeed.